Your brain is retiring its own stem cells, and by the time you hit your 60s, the factory that builds new neurons has practically shut down. But a team out of Singapore just found the protein responsible for that shutdown, and when they cranked it back up, those retired stem cells went right back to work.
Wait, hold on. Back to work as in actively dividing and making new neurons again.
That's what the data shows. The study dropped February 12th in Science Advances from researchers at the National University of Singapore. The lead author is Dr. Liang Yajing, working under Assistant Professor Derek Sek Tong Ong, and the protein they zeroed in on has a name only a scientist could love, cyclin D-binding MIB-like transcription factor 1. Thankfully, we can just call it DMTF1.
Okay, so before we get into what DMTF1 does, let's set the stage. Why do our brains slow down in the first place?
Great question. So deep in your brain, you have these neural stem cells, or NSCs. Think of them as the seed stock for new neurons. When you're young, they're constantly dividing, producing fresh neurons that plug into your learning and memory circuits.
And as we age, they just stop,
They go dormant, almost like they're retiring after decades of service, and there's a very specific biological reason for that. Telomeres.
right? The protective caps on the ends of your chromosomes.
Every time a cell divides, those caps get a tiny bit shorter. It's like a biological countdown timer, and after enough divisions over a lifetime, the telomeres get so short that the cell basically says, I'm done. I can't safely divide anymore.
So the stem cells aren't dying necessarily, they're just refusing to work.
Correct? They become quiescent. They're still there sitting in your brain, but they've stopped producing new neurons. And that's when you start noticing the cognitive decline. The memory gets foggier, learning new things takes longer, processing slows down.
Okay, so that's the problem. Where does DMTF1 come in?
So the Singapore team wanted to figure out the molecular machinery behind this shutdown. They took neural stem cells derived from humans and also worked with mouse models that mimic premature aging, and they ran genome binding analysis and transcriptome profiling to map out which genes were active and which were silent.
And DMTF1 popped out of that analysis.
It was front and center. Here's what they found. In young, healthy neural stem cells, DMTF1 levels are high. In aged neural stem cells with shortened telomeres, DMTF1 levels were significantly reduced. There's a clear correlation between aging and losing this protein.
So it's not just that the telomeres shorten, the telomere damage is actually draining this specific protein from the cells.
Right? And here's where it gets really exciting. When they artificially restored DMTF1 expression in those aged stem cells, the cells started dividing again. They regained their ability to regenerate.
Even with the short telomeres still being short,
That's the wild part. The telomere length didn't change. The telomere stayed short, but DMTF1 essentially found a workaround. It's like the cell's countdown timer is still at zero, but DMTF1 overrides the retirement notice and says, get back on the line. We're making neurons.
that's fascinating. So it's not reversing the aging of the telomeres themselves, it's bypassing the consequences of that aging.
Precisely. And they figured out the mechanism too. DMTF1 is a transcription factor, which means it's a protein that controls which genes get turned on or off. Specifically, DMTF1 activates two helper genes called ARID2 and SS18.
What do those do?
They're part of something called the SWI-SNF chromatin remodeling complex. In plain English, they loosen up tightly packed DNA. See, your DNA doesn't just float around loose in the cell, it's wound up tightly around proteins called histones, like thread on a spool.
And when it's wound up tight, the genes can't be read.
Right? The cell machinery can't access those genes, so what ARID2 and SS18 do is they unwind that DNA in specific spots, exposing growth-related genes, and those growth genes then activate what's called the E2F pathway, which is the biological cycle through which new neurons are actually created.
So let me trace the whole chain. Telomeres shorten, DMTF1 drops, ARID2 and SS18 go quiet. The DNA stays wound tight. Growth genes can't activate, and the stem cells stop dividing.
You just summarized the entire paper, and the intervention point they identified is DMTF1. Boost that one protein, and the whole downstream cascade kicks back into gear.
Now I have to ask the obvious question. Can they do this in a living brain, in a person?
Not yet, and the researchers are very upfront about that. This work was done in vitro, meaning in cell cultures in the lab, plus some mouse model experiments. Professor Ong himself said the study is in its infancy.
But they have a roadmap.
They do. The next step is testing whether boosting DMTF1 can increase neural stem cell numbers and improve learning and memory in living animals with shortened telomeres and natural aging, and they want to do this without raising the risk of brain tumors, which is a critical safety concern.
Yeah, let's talk about that because whenever you hear we made cells divide more, the immediate worry is cancer.
Absolutely. That's the tightrope walk with any regenerative therapy. You want cells to divide enough to restore function, but not so much that they go rogue. The fact that the telomere stays short, even when DMTF1 is boosted, might actually be a safety feature here. Short telomeres are one of the body's built-in tumor suppression mechanisms, so in theory, you're getting the benefit of renewed stem cell activity without removing one of the brakes that prevents runaway growth.
That's actually a really elegant balance if it holds up.
It is, but it's all theoretical at this stage for humans. The long term goal for the team is to identify small molecules, basically drugs, that can safely stimulate DMTF1 activity to rejuvenate aging neural stem cells.
So we're talking about a potential pill or treatment that could keep your brain producing new neurons well into old age.
That's the dream, and it's important to put this in context. The debate over whether adult human brains even make new neurons at all has been going on for decades. It was only relatively recently that researchers confirmed adult neurogenesis is real, that there are nurseries in the brain, particularly in the hippocampus, that keep producing neurons throughout life,
But that production slows way down.
dramatically. And this study gives us a specific molecular target for why it slows down and how we might speed it back up. That's what makes it significant. It's not just we found something interesting, it's we found a specific protein, we mapped its entire downstream pathway, and we showed that restoring it reverses the problem in aged cells.
Who funded this research?
It came out of NUS Medicine, specifically the Department of Physiology and the Healthy Longevity Translational Research Program. Singapore has been investing heavily in aging research, which makes sense given their rapidly aging population.
So what should people take away from this? Because I know listeners are going to hear reverses brain aging and start Googling where to buy DMTF1 supplements.
Yeah, please don't do that. This is basic science, not a product launch. There's no DMTF1 supplement. The protein is a transcription factor that works inside cells at the genetic level. You can't just eat it.
What about the broader implications, though? Is this part of a larger trend?
Absolutely. We're seeing an explosion of research into cellular aging mechanisms, telomere biology, senescent cell clearance, epigenetic reprogramming. This DMTF1 finding slots into that bigger picture. It's one more piece of the puzzle of why we age and what we might eventually do about it.
And for our brains specifically, the stakes are enormous. We're looking at a future where diseases like Alzheimer's and other forms of dementia could potentially be delayed or mitigated if we can keep neural stem cells active longer.
That's the hope. Dr. Lang said it best. Our findings provide a framework for understanding how aging-associated molecular changes affect neural stem cell behavior and may ultimately guide the development of successful therapeutics. It's early, but the foundation is solid and the pathway is clear.
Your brain's stem cells haven't given up. They're just waiting for the right signal, and now we know what that signal might be.
DMTF1, remember that name?
Thanks for listening to Things I Know Nothing About. If this blew your mind, share it with someone whose brain could use a little rejuvenation.
See you next time.